My name is May-Britt Tessam. I'm a researcher and an associate professor at the Department of Circulation and Medical Imaging at NTNU. And my research is focused around prostate cancer and molecular multi-omics technology on clinical tissue samples. The ProstOmics project is an ERC starting grant. The goal of this project is to identify biomarkers and understand molecular mechanisms on how prostate cancer becomes aggressive and life-threatening. So ProstOmics is based on analysis of larger patient cohorts of high-quality frozen prostate tissue that is donated from surgery of patient with consent. And we drill frozen and targeted tissue samples from several places within the prostate to study heterogeneity within the tumor, around the tumor, and also in normal appearing cells. So from each selected sample, we cut very thin sections of the core, which is used for the various omics analysis and histopathology. So we have recently focused on omics method that provides spatial information within a tissue. We want to understand the molecular mechanisms within the tissue microenvironment. By doing spatial analysis, you can go deeper in your analysis and you can identify biomarkers that haven't been identified before. So an important method we use in the ProstOmics project is multi-TUF mass spectrometry imaging, where we receive molecular maps of metabolites and lipids and peptides almost down to cell level. So this method uses laser ionization directly on the tissue, which is coated with a specific matrix, which is sprayed in layers directly on a tissue section. The specific machine can be extremely fast because the laser shoots rapidly over the tissue surface, and one small tissue sample can be finished within minutes. We also use spatial transcriptomics, where we can see gene expression of thousands of genes like a map on our tissue samples. We also use different microscopy techniques like confocal microscopy, where we stain different compartments or specific proteins of each cell in the prostate tissue to get really high detailed 3D images of the selected proteins. So by integrating the data from the multi-omics results, we aim to understand more on the molecular mechanisms that separates aggressive from slow-growing types of prostate cancer. It's especially exciting because I designed the project myself. We have built up this activity for years here in Trondheim, and we also have excellent collaboration with the St. Olav's Hospital and the Biobank 1, where we have all the high-quality tissue. I hope that the patient first will benefit from this, and also good biomarkers will benefit the clinicians that have better tools to give the right treatment at the right time. And I also hope that the society in the whole can benefit from this by reducing overtreatment and reduce the costs and the resources needed for treating patients.